So you're tired and you want to take a snack, and then you take out a kiwi because it's healthy. Well, what if a kiwi wasn't healthy? Okay, what about an orange? Okay, but what if the orange also wasn't healthy? Okay, what about bread for a sandwich? No, that's also not healthy. These are all foods that are loaded with sugar. And when I mean loaded, it doesn't mean they're actually bad. It's just they have a lot of sugar in them. And so for some patients, especially like type 1 diabetes, which comes often from a very young age, they're restricted from those foods. But what they do get is a lot of pills. And these medications often have side effects. For the longest time, we've been treating diabetes with lots of medications, especially one we know as insulin. But the main question is, are we really solving the fundamental problem here? Because there may be a genetic problem at its very core that may be the cause of all of this. But what happens if we can get rid of that genetic problem in the first place? Hey there, my name's Ahash and I am a 16-year-old genetic researcher at the Knowledge Society. This year I worked on a hypothesis to slow down the effects of type 1 diabetes using CRISPR-Cas, wild-type adeno-associated viruses, and also pancreatic organoids. Today I'm going to be talking about the same hypothesis, but I'm going to be breaking it down into a few sections. First, I'm going to be talking about what type 1 diabetes is, then going into how it relates maybe to the CERT1 gene. Then I'm going to be talking about CRISPR-Cas systems, adeno-associated viruses, and pancreatic organoids. Let's get into it. Imagine you were doing your thing, and then someone came and started destroying everything you created. Yeah, I would also be confused. But this is the reality for a type 1 diabetic patient. So type 1 diabetes is an autoimmune disorder. At its very core, it's basically where our immune system is targeted towards insulin cells that are produced by the beta cells. This is basically where you may have mutations, maybe it might be a genetic cause, and where our beta cells produces these insulin cells, they end up being attacked by cells like macrophages and T cells. The problem with this is that insulin is actually very important to our body. For example, when you eat, let's just say a kiwi, and you chow it down, it goes into your small intestine, which gets broken down into our sugars. Now when they get into our bloodstream, they have to be either one, utilized as energy, or number two, they have to be stored as energy. And insulin helps in the two processes that aid in the end result. The first up is glycolysis. In this process, essentially insulin can be a key, I guess, and here it's able to unlock our body cells to use glucose, and that way the glucose can be immediately used. But sometimes we don't need the energy immediately, so lipogenesis occurs. This is basically where we convert that glucose into fatty acids, which gets stored as fat, and this is also where insulin can come in. Insulin is really key because without these two processes, we often end up with maybe high blood sugars when we eat, or when we don't eat and we're using that energy, we end up with low blood sugars. And these may seem like short-term losses, but actually they can lead to long-term diseases. When we're looking at type 1 diabetes more particularly, there's one key correlation here. CERT1, it's often found to be a repressor for the UCP2. That's basically the uncoupling protein 2 in our beta cells, and they end up helping with insulin secretion. So there's also a study that suggested that it may be the cause of type 1 diabetes. They found in residue 107 in the exon 1 of the CERT1 gene, there was a T to C mutation. Now what that means, instead of having a T, they end up having a C, and that correlated with an excessive amount of nitric oxide, chemokines, cytokines, and these all end up being signalers for the immune system, and that triggers an immune response against insulin cells, which are produced by the beta cells. So this study may show a potential here to track down the causes of type 1 diabetes, and that's exactly where CRISPR-Cas systems can come into play. The way way it works is that usually you start off with an sgRNA. Now this sgRNA, also known as a guide RNA, can help guide what we know as the Cas9 endonuclease to the site, so it's able to bind to the target DNA sequence. And then we use a PAM, which is a protospacer adjacent motif. That protospacer adjacent motif has the ability for the Cas9 to differentiate whether the guide RNA or the target DNA is the one we want to actually target. And the Cas9, which is basically like a pair of scissors, has the ability to just cut our DNA sequence just like that. We can then use what we know as a homology-directed repair to repair the sequence with the donor template, and that enables us to potentially erase the mutation once and for all. In order to get the system delivered, we can actually use adeno-associated viruses, and these might be the answer. Adeno-associated viruses are basically certain vectors. We know them as viral vectors. They're really special because they aren't as harmful for humans since they're genetically engineered, and they have the capability to not replicate inside the cell and cause damage to our bodies. They're also a bit better than retroviruses because they're safer, and because they have inverted terminal repeats at the end, these have the ability to better transfect the DNA. So it's a really optimal viral vector, which is why I chose this to include in my hypothesis. In order to actually simulate this and make sure that we have the most accurate answer here is really the utilization of pancreatic organoids. For organoids, they're basically cultured stem cells, and this can be from iPSCs. iPSCs, which are induced pluripotent stem cells, are basically specialized cells, and when we use genetic engineering, we can actually manipulate them to be back into the stem cell state, and that way we can use them to specialize into other cells. If we're looking at, for example, an ex vivo, so an in vitro utilization of these pancreatic organoids, we can actually culture them with the correct combination of growth factors like B27, and that way they can actually almost mock a pancreas, more specifically the pancreatic islets. And the reason it's really important here is because mouse models may not be the most accurate answer. The problem with mouse models is that we think that they're actually very accurate, but they aren't because the differences of the anatomy of pancreatic islets. If we take a look at a mouse model, you kind of see that there's a lot of red, and what that means is that there's a bunch of different types of cells, like alpha cells, delta cells, PB cells, so many different types of cells, but inside is really where the beta cells are. And so it kind of surrounds it like the core of a corn. But if we're looking at a human model, it's very different because you have an equal, almost equal distribution of different types of cells, including the insulin cells. What that means is that the sensitivity to glucose could be drastically different. And so what we want to do here is make sure that we have the most accurate model of these specific CRISPR knockouts. And so by using pancreatic organoids, they can pose a whole new future because we can test them more accurately, and this will ultimately influence the efficacy of our experiment. Ultimately, I want you to leave this video with knowing how we could potentially utilize these CRISPR-Cas systems to treat type 1 diabetes. If you're more interested, you can check out the full report on my personal site or the article I wrote on it. Thanks for listening.